Well, thank you, Professor Cotter, for inviting us over to your home to talk about your experiences in your career in haematology. Obviously, it's been 60 years now of the British Society of Haematology. So, would you like to tell us a bit about your work over your career?
It's a pleasure to meet you in my home. I'm Finbarr Cotter. I'm a haematologist at Barts Health. And I've been in haematology for not quite 60 years, but over 40. And it has been interesting times. When I first started, we were at a time when leukemia was just beginning to be treated for the first time. Childhood leukemias were a killer. And Judith Chessel's at Great Ormond Street was just beginning to treat these leukemias with great success. And we are now in a totally different area. We have immunotherapy, we have biological therapies, we have genomics, we have a whole range of different elements that we can contribute to managing our patients. So exciting times, and that's just malignancy.
So when you look back 40 years ago when you were thinking about your career, what made you consider haematology?
Serendipity, really. I would say now, why didn't I do haematology? It was the obvious thing to do. I think the thing which really struck me more than anything else as really exciting was one weekend when I remember being on, and a lady came in who had a hemoglobin that was below 20. Yeah. And I went along with the senior registrar, as you did as a junior doc then, who did the bone marrow. We went to the lab, you stained the slide yourself. He came out and he said, "This lady's got pernicious anemia." We went in, she was given some B12 and she wasn't transfused. Then a day later, another lady came in who had complained that she was short of breath when she was out shopping. Again, she had a hemoglobin which was around about the 2 level again. And so he did a bone marrow, went off, did some stains, and said, she's iron deficient, and she was started on iron. About a week later, the 2 ladies were skipping around and they had a total hemoglobin that I think came to probably around about 80. And I thought, this is quite good. It's kind of do-it-yourself medicine. And I think that was really what got me interested, the fact that you were kind of self-contained. You had both the laboratory and the clinical aspects under your control. Whereas most of the rest of medicine and surgery, you are reliant on the pathologist to tell you what to do. So yeah, I thought this has to be the right thing to do.
I see similar things. That's the reason why I went into haematology as well, that ability to be able to actually take a sample, go to the lab, diagnose and then treat it all yourself is definitely a major benefit in our field. When you look back, what's the sort of main successes or highlights of your career, do you think?
Well, I think there are a few things. There are successes and there are failures. I think I'm extremely glad that I went off to do a PhD. I was involved in, I actually administered the chemotherapy to the first patient at what was the London Hospital, now the Royal London Hospital for acute leukemia as a junior doctor, and the patient did incredibly well. And I thought, actually this element of new medicine and something that I'd been taught in medical school was a death sentence, actually had something that could make a difference. And during my training, I felt that the word oncogene then appeared. I read about it, and the whole concept of DNA and coding seemed to be relevant, and it seemed to me we were doing a lot of treatment without understanding the mechanism of the disease. Maybe if we knew where the code had gone wrong, we might be able to do better. And so I went to the ICRF as a research fellow, and in fact based at Barts for my clinical work, and at ICRF where the Genome mapping project, there was a chap called Hans Lirac who was an Austrian guy who had been employed by the Imperial Cancer Research Fund, now CRUK, and they put a lot of effort into mapping the human genome. And I was involved in flow sorting chromosomes from a chromosome mixture in order that they could be turned into what we called libraries of DNA, which the sequencing took place from. And I learnt to sequence, I learnt what DNA was about, I learnt to put DNA into bugs, and to really learn a lot of the tools as doing my PhD around DNA. And the genome was mapped. We now are mapping these things very, very readily in a very short space of time at minimal cost compared to the huge cost in doing that. But I work particularly on lymphomas and around the 14-18 translocation in follicular lymphoma. It got cloned. At that time, I was doing sequencing around that, and it looked as though then we discovered the function of that protein that came from there was a survival protein, and that these cells couldn't die, these malignant cells, because they didn't have the ability to turn off a survival protein. And so I went on to develop an antisense oligonucleotide, which I was then able to relatively easily find a company to go out there to manufacture, do the work. David Cunningham at the Marsden was prepared to allow us to use it on the patients. And we did the first trial in the world silencing a gene, the BCL2 gene, to good effect in patients with follicular lymphoma. Now, BCL2 we now have with venetoclax and as one of the mainstays for a lot of cancers and as a form of treatment. We went on then to do some more extensive trials, and I feel that was something I'm proud of. There are patients who were not going to survive, who did survive in that initial study, and that was really exciting because it was the first time we can show using a genomic therapy you are going to change the course of disease. And really the last 20 years has been about bringing that through. Immunotherapy has come of age. Again, that took 20 years to come through. Most discoveries take a long time to get there, but to me, that's something I'm proud of. But probably the proudest thing is actually bringing through other hematologists, younger people, to make them as enthusiastic as I feel about it.
Obviously, as you mentioned, there's been a lot of advancement in the past 20 years in genetics and immunotherapies. Where do you think that treatments are moving over the next 20 years or even?
I think we're moving towards cancer and malignancy becomes more of a chronic disorder as opposed to this acute, do or die disorder. Where we use poisons and radiotherapy or magic rays to burn the things out or get the knife out, to something that we can manage with a daily tablet, for example. And we're learning to control these different elements so that we can then use biological medicines, which we have learnt about by dissecting the biology of the disease, so that we can control the disease even if we may not always cure it.
And how do you see the British Society of Haematology's role in the coming years? How do you think that they can help both in the development of new trainees and encouraging people to come into haematology as a specialisation, and also to help with the advancements that you've just been talking about?
I think as a professional body, as a professional society, it has a pivotal role in representing the haematologist. I think there are issues around colleges where they represent swathes of physicians, swathes of pathologists, and have to take into account all of the different types of pathologists and physicians around. I think that we understand our own business, and what is important is that you involve people in the field of haematology from a very early age, by which I mean from trainee level upwards, so they are involved in the process. because that makes you vibrant and it gives you continuity.
And what would you say to someone who was looking, so a junior doctor who was thinking of a career in haematology? How do you think you'd, I mean, you've done a very good job so far, but I mean, how do you think you would sell it to them and encourage them into the field?
There is no other specialty that really crosses all of the fields and is as interesting, with a whole community of people who are absolutely fascinated in their different areas. And it covers everything. Why not haematology? It is a model for all disease.
Do you think the workforce is changing? Do you think that as you were saying about subspecialising in haematology, do you think there'll ever be a change in the way that we train haematologists?
I think there's inevitability that we become subspecialised. It's a question of whether you want to have a broad training first of all. If you are running a government and a healthcare system, you want to get a doctor to the point where he specialises and does what you need as quickly as possible. Yep. So I can see the pressures on that. But at the same time, you may ultimately be more valuable and more flexible, because if your whole area of specialty changes, so for example, you're a bone marrow transplanter and Gleevec comes along for chronic myeloid leukemia, you have to change yourself from being a transplanter to somebody who understands the biology of disease, and we need to adapt. If you haven't got a broad enough base, you may find it more difficult to adapt because you've been put into slots where you get very stuck in those areas. However, the trick is that we should be looking at having allied healthcare professionals having much broader roles to support what we're doing, and that you have the consultants who are there who are able to take a broader overview and to be looking at more complex issues and to feed into a system so that you're able to provide that healthcare without escalating the costs beyond what we can afford as a nation. And that applies to all countries. I mean, the US has a big challenge that they're nearing 30% of their gross national product just on providing healthcare. Well, your economies don't thrive well if you're spending all on just keeping yourself alive and healthy. And I think we have a real issue around making sure that we get our workforce right, and that they have the training still is a broad training, so that you have that broad spread, which allows you then to subspecialize into what's appropriate. But if things change, you can regroup, and you will still be extremely useful because you have the tools and the equipment to allow you to contribute into running specialties.
And often, I mean, as juniors, we're often sort of advised to find a mentor or someone that you can go to who's senior, that you can ask advice and career pathway guidance, questions, ask them things about that. Do you think that's a good thing, that you should try and find a mentor? And for you, for example, did you have anyone that you looked up to or spoke to?
I have had a number of people who I have looked up to who've been great mentors, not always necessarily hematologists, but I think that having people that you can discuss things with who have your best interests at heart is important. And to me, the mentoring and the number of people who I've tried to encourage and to bring through, and the journal in part is also a means of mentoring. And we run workshops to try and help educate people around how to write a paper, how to do things. It's having somebody that you can kind of pick up the phone and say, what do I do here? Because careers are difficult. And sometimes you need a point in the right direction from somebody who hasn't got any reason to be biased about this, that, and the other.
Okay. Well, I mean, thank you very much for inviting us here to talk through your experiences and the insights into your career, which is still ongoing. And, hopefully the haematology will continue to progress, as you've mentioned, and the BSH will play a pivotal role in that.
It's been a fantastic career. It continues a bit more, but the nice, encouraging thing is seeing the future. Yeah. and the future is continued over those decades. They've been coming up and they will continue to do that. Thank you very much too.
Oh, well, thanks. You've certainly made my job as an interviewer very easy.